So we're going to do Q&A. We're going to, let's see, Dave is going to come back up to the stage, and the techs are going to put some mics out. I've got this one, I guess I'll do it. So why don't we line up here like before, and I'll just hold on to the mic a little differently this time. Great. So I guess you're the first question. Who's it for?
My name is Mario. I'm a physician from Canada, and my question is for Dr. Nichols. Can you just comment on the pharmacology of LSD in the context of possible serotonin syndrome on patients that are on SSRIs, and whether the long-term binding of LSD to the 5-HT2 receptor could potentiate that, and what implication that might have on the psychedelic effect on somebody on an SSRI, and the clinical effect of a possible serotonin syndrome. Really appreciate your thoughts. Thank you.
Was that one question? The serotonin syndrome is generally related to an excess production of serotonin. And of course, LSD activates 2A receptors, and there's actually a feedback mechanism that would decrease the release of endogenous serotonin. And I think it's been shown if people have been treated with an SSRI, which increases serotonin, it actually downregulates those receptors, and LSD would have a blunted effect. So I don't know that there's any evidence for the serotonin syndrome with LSD per se, and I don't know that it wouldn't increase serotonin, which is generally what drives the serotonin syndrome. Hope that answered your question. That's about the best I could do on that one.
Hi, my name is Balázs, and my question is about the difference taking LSD intravenously or orally. There was some discussion about that, and what I would be interested in is that the subjective experience of the people who take the same amount of LSD orally or intravenously, are they the same intensity of the experience, or is it more intense if people take the same amount of LSD intravenously? I wonder what's the case there.
Thank you.
Okay, can answer that. We did some studies intravenously more than 10 years ago and compared different doses and the time, the process, how long it lasts. If you give it intravenously 1 or 2 milligrams, we did at time up to 3 milligrams, lasted not very long, about 15 to 30 minutes, and the whole thing is over, and it's immediately you are on the trip. And there was always a discussion. We did the same 8 of the subjects also had it orally, 15 to 20 milligrams. It was a little bit more enfolded orally. People had much more time to go into depth. We did it in a relaxing situation. Even we had to take blood samples for pharmacodynamics and kinetics. But it was, according to my understanding, we didn't do a lot of comparison. 8 subjects is not much to say. It was more profound. It was my experience. But I don't want to, it's a cautious thing because you have a different mechanism and you have time. You can deal with the stimuli that come up or the memory and all that stuff. It would need more studies to go into that, more detail.
You're referring to psilocybin.
Yeah, just psilocybin. LSD, we don't have done intravenous studies, but they are published, and that's something I have to look at a little bit closer. There are studies that showed relatively fast onset within 20 minutes, so I'm not sure.
I spoke with Ben Sessa last summer. He was one of the subjects in the Imperial College study where they gave 75 micrograms intravenously, and he said it was really strange because he also was a psilocybin subject, and you give him psilocybin and you rocket it out of this cannon in a few seconds. And he said the LSD took about 30 or 40 minutes to come on, and it was almost as if he'd had an oral dose and it lasted about the same length of time. So that really piqued my interest in terms of why is that, because it's so different from any other intravenous drug, and I think it's related to the fact that it takes a long time to actually get into the receptor. And then once it's there, and it was, I think IV, someone can correct me, I think it was 75 micrograms intravenously in the Imperial College study, so they would sort of know what 75 micrograms orally would be, and he said it was about the same as oral, he thought. But that's an N of 1, somebody can correct me.
My name is Link, and Dr. Nichols, thank you for the compelling explanations of the potency and duration of the LSD. It got me thinking that that could possibly lead to pharmacological techniques for changing the duration of an LSD experience, maybe terminating it early, God forbid, elongating it, because of the way that the new knowledge of how it interacts at the receptor site and changes the structure of the receptor. Perhaps there could be other techniques to change the structure of the receptor while the LSD is in there to cause it to be released sooner. I'm just wondering if you have comments on that.
Yeah, there probably isn't a way to do it in humans. You could make a transgenic mouse where you put in the human 2A receptor and mutated that leucine to an alanine and look at the duration of effect in the mice, and that would give you some idea of whether these in vitro effects translated. We have made lysergamides that have a shorter action, but nothing is like LSD. So I don't know really how you would do that.
Thanks. Yep. And I just thought that a useful technology could be a way to completely abort the LSD trip.
Yeah, the question I raised with Franz yesterday was they pre-treated all their subjects, with the LSD subjects, with ketanserin. They pre-treated, so it's in the receptor. The question is, if LSD is locked in the receptor, if you now give ketanserin, can you then abort the trip? And I think that's a very interesting question.
We could do it for psilocybin, but no, we didn't do it for LSD.
Yeah.
If you have psilocybin in the body and we injected ketanserin, it's gone in 5
Yeah.
minutes.
Psilocybin probably doesn't get trapped, though.
Yeah.
It's
I don't know.
fast in, fast out.
Thank you.
Hi, I'm Will. I just had a question for Dr. Nichols. Would you attribute, if you were going to take a stab in the dark, the qualitative differences between the experience of your lysergamides like ALLAD, ETHLAD, maybe ALD52 and LSD to, is there a different affinity for recruiting the beta-arrestin or like a different GCPR complex? Or because it seems like from reports that like for a roughly equivalent microgram to kilogram dosage that there are very different subjective effects in terms of the ratios of visual hallucinations to like brain fuckery or whatever that quality is. What would you attribute the differences to, and or did you do like an arrestin side by side between those in the same way that you did with LSD? Thanks.
Yeah, some of those studies are underway. Undoubtedly, did you use the term brain fuckery? LSD is probably optimized for brain fuckery. But you know, there is some work in progress with ETHLAD, which is actually more potent than LSD. And you'll learn more about that, and I can't speak to it because it's work in progress. It hasn't been published. But there are differences, and I would point out that LSD is not a full agonist. When it binds to the 2A receptor, you only get 35 or so, 40% activation. And there are some of these other analogs that are actually full agonists. So the signaling capability and the extent of signaling and the pattern is probably different for all these. So LSD is really a unique molecule.
And so, but that would be for all like G protein different signaling and the beta arrestin downregulation of the receptor, or both.
There would be a different signaling bias probably.
And so we would expect that like the tolerances may be different if like the arrestin is more or less regulated than later on fewer receptors expressed. Or?
Yeah, I think the tolerance would be similar. I don't think that's a subtle thing. That's a general phenomena. But in terms of the balance of some of the other signaling pathways, you might get some qualitative effects. But there's no one going to fund a big study comparing the qualitative effects of ETHLAD, ALLAD, LSD, et cetera. It'd be interesting, and maybe someday, and then to be able to correlate it with signaling patterns in the cell. But interesting question, but beyond the state of the art now.
Thank you for your question.
Hi, I'm Paul, and I'm curious if, with respect to latency and duration, if you've made any simulation probing of other long-acting psychedelics like DOM that have much longer latency periods and much longer duration.
You know, the pharmacokinetics has not been studied on hardly any of these. I mean, There's a paper here from Wisconsin about pharmacokinetics of psilocybin, and there's a study from Felix Hassler, I think, on pharmacokinetics. All of those drugs have different hydrophobic characteristic. And so when they partition into the brain, assuming that the PK is the same, they're going to partition in different proportions and in different ways. Very hydrophobic things partition differently than things that are less hydrophobic. You know, the NBOME compounds, for example, are not orally active in most people because on the first pass in the liver, they're so lipophilic, they are trapped in the endoplasmic reticulum, and P450 OD methylates it. So you have variations of how much would be metabolized on first pass, and then how much is actually absorbed, the fraction absorbed into systemic circulation. So those would be unique for each molecule. You change from, you know, methyl and DOM to methylthio to bromo to iodo. It's going to change its partition coefficients. It's going to change the relative metabolism, the fraction absorbed. So there are so many factors involved in that. And probably the same person taking the same drug at the same dose on Monday may have a different effect on the following Monday, just because in changes of the brain, dynamic changes. So that's very difficult to really pin down because there's so many variables.
Thank you.
Hi, my name is Caitlin. I had a question. About a year ago, I saw a paper published talking about the mechanism of ayahuasca, but they talked specifically about this receptor dimerization between the 5-HT2A receptor and metabotropic glutamate receptors. Do you have any awareness that LSD is doing this as well? And if so, do you think that there are both direct and downstream effects of stimulating these glutamate receptors that might be, upregulating neurotrophic factors?
So that's a somewhat controversial finding. It's clear that there's a functional interaction between glutamate and 5-HT2A receptors. The original paper suggested that they formed a heterodimer, the two interacted. Those studies were done in mouse somatosensory cortex, not in prefrontal cortex. Somatosensory cortex has a lot more of the metabotropic glutamate receptors than prefrontal cortex. The other issue to keep in mind is that serotonin 2A receptors are expressed postsynaptically, and metabotropic glutamate receptors are expressed presynaptically. So the possibility of them forming functional heterodimers in prefrontal cortex, in my opinion, is very low. But there is a functional interaction. You can block behavioral effects of psychedelics by blocking metabotropic glutamate receptors, and in drug animal studies, clearly there's a glutamate effect. They release glutamate, it activates those receptors someplace, but whether it's a functional heterodimer, I think I'm skeptical, and I know an awful lot of people just don't believe that really happens. But there is an interaction. Clearly glutamate is important for the action of psychedelics.
Thank you.
My name is Poncho. My question is similar to Paul's regards residence times. You indicated that the residence times for LSD and the 5-HT2A were about 8 hours, which is pretty similar to the length of the effect of the cognitive duration. Would you expect correlations across others, especially in situations where the blood plasma concentrations deplete significantly faster than the residence time?
Say that again.
So the question is would you expect correlations of residence time or latency to the duration of the effect for other classes of psychedelics besides LSD?
Mescaline is a long-acting compound. It's a fairly polar compound. No one has ever looked, as far as I know, at the kinetics of these things are difficult because you really need to get radioactive mescaline or radioactive whatever to do the study. I suspect the duration of for some of these others is at least partially related to residence time in the receptor. But LSD is also not very potent. It takes a large dose. Its log P is around 1 something. So you have to really put a lot of it in. And then it's not metabolized very quickly. So there's a lot of other factors as well. LSD binds to a lot of other receptors, D2, D4, some of the others, and they don't actually have that same loop that traps it in there. So for LSD, you may be activating the 5-HT2A receptor for a long time. LSD may be activating, say, D2 receptors, but for a shorter time because the efflux is faster. So you're going to get differential effects depending, even with LSD itself, on how long it resides in those different classes of receptors.
Thank you.
My name is Lorenzo Pasquini. I'm a postdoc in neuroimaging, and I have a question to Mr. Vollenweider. And I found it particularly striking that two networks which I've found to be modulated by the action of psychedelics, namely the default mode network and the salience network, are also differentially targeted in two types of dementia, namely the default mode network in Alzheimer's disease, where you have the typical loss of yourself, memory impairments, and the salience network on the other side, for example, in frontotemporal dementia, where you have these striking social dysfunctions, loss of empathy, and so on. So I was wondering if you could discuss this interesting overlap and also maybe speculate on how the current research on psychedelics can help us to understand dementia or also to treat dementia at some point.
Now you have to keep in mind that this model here is just a temporary short change for 2-3 hours. So basically on that acute model you can maybe get hints what part of the default network or the midline structures who are responsible for self-other or forming self-other boundaries are involved, but I'm not sure whether it really says a lot about Alzheimer's, because I'm not sure whether they have a disturbed sense of self, whether they still experience themselves as a whole, or whether it's just the memory that they constantly forget who they are. So I don't think it's logically the same process.
Okay, thank you.
Hi, my name is Nick. This was a massive amount of data that was presented, and I was wondering if you could make your slides available, or if there's like a reading list of key journal articles that you could make available
sure.
to the participants.
Most everything is published, and there are downloadable PDF PowerPoints on our homepage now. You can look at different themes. You have about hallucinations, basis of self, neurochemistry. Just jump in and download that stuff.
Okay, thanks.
Hello, my name is Kevin, and I just wanted to ask an open-ended question of sorts. An open-ended question of sorts. So given that we have all this new research on connecting the subjective aspects of psychedelic drugs to some objective measures like brain blood flow and receptor activation, what do you think is the next major step in connecting the subjective experiences of subjects to the objective science of what's happening in the brain? And how can we get closer to understanding consciousness with these research efforts?
What I think is discussed that a lot with good friends in consciousness research, like Christoph Koch, the Paul Allen Institute, Tononi in medicine. We have to come up with, Tononi suggested something, other people also, with a theory that explains how invariant phenomena drive the brain. People look now at the brain and make an idea what constellation of neural activity could be responsible or enable a psychological phenomenon, but that's the wrong way. You have to come from the phenomena and look what goes in parallel in the brain. We have to have a better understanding of the phenomena and what phenomena are invariant, and it's hard to crunch down the self, like a feeling of agency. They can make some tests. It comes into cognitive science. It's not what we really want. We want the phenomena. But there's also a philosophical discussion whether you ever can qualia reduce to neuroscience, because the experiential sense or feeling of red is not coded in physics, in the language of physics. I don't know whether you understand that, but it's not possible.
It's a very complicated function. But I think that eventually we may be able to get closer to connecting the brain.
You get closer to understand how these things go in parallel, that's certainly, but ontologically not. What's the real basis? What's really consciousness? We think it's an information coded by material and energy fields. So, and that is what has been suggested recently, to understand the so-called code in terms of information.
Thank you. Can I make a general comment? Everybody here that's probably not 100 years old, is living in a really exciting time. You probably know this. Research was stopped in basically 1970, and the kind of things you're hearing here in Imperial College, this is half a dozen groups in the entire world. But people, young people especially, I see them all the time, they send emails, how can I get into this field, how can I do this? People are going to medical school. So you're going to see a lot of this, and if we can get the governments engaged in funding this at a fundamental level, you're living at a time where we're really going to begin to see some things that, questions that people have asked forever. Who is man? How do we work? What's consciousness? So just as a general comment, I think it's a really exciting time. We're doing as much as we can, but there need to be a lot more people, a lot more funding, and the questions are out there. So you're going to see some of these in your lifetime, certainly.
You know, I think we've run out of time. We want to take our break. So I'm going to leave it at that, Dave. That's a wonderful way to close it. Dave Nichols, Franz Vollenweider, help me thank them, please.